In our last video for this unit, we're going to look at nucleic acids, and this would be the fourth macromolecule that we mentioned a ways back. Remember, they were carbohydrates, lipids, proteins, which includes enzymes, and nucleic acids for the fourth. Now, the elements that make up a nucleic acid are carbon, hydrogen, and oxygen. Those three we have in all of them: carbohydrates, lipids, proteins, and nucleic acids. We have nitrogen. Nitrogen was only with proteins, and nucleic acids have nitrogen. And now nucleic acids has its own special element, phosphorus, which is not found in the others. Some examples of nucleic acids. You may have heard of DNA. DNA stands for deoxyribonucleic acid. And you can see a picture here of DNA. You also have RNA. Notice that the Na stands for nucleic acid, so RNA is a nucleic acid, stands for ribonucleic acid, and it looks like this one-stranded object here. Some jobs: they store and transmit genetic material, and you may have heard of people asking for DNA testing. So this little boy is saying, "You're the mother and those are your children? I'd like to see a DNA test." So DNA testing, we know, can kind of help us decide if a child belongs to a certain parent or set of parents. And that's because your DNA stores this genetic material, so we can see how similar DNA is from parent to child. It also helps in making proteins, and that's more of the RNA job. Has a direct connection with helping to make protein. With each one of these, we had polymers, except for lipids. And each one of our polymers had a monomer, something small that they are made up of. And for a nucleic acid, the monomer would be a nucleotide. And a nucleotide is still pretty large. It's actually made up of three parts itself. So if you see a picture, you can see them grouped here together. And they'll show up on your screen here in just a minute. I'm just highlighting them for you. But those are the three main pieces. But this one object, all of that, is a nucleotide. So the parts that you would have is a nitrogen base, and we'll talk about the nitrogen bases. There's 4 different possible nitrogen bases. A 5-carbon sugar, and depending if you're using DNA or an RNA, the sugars are different. And then you have a phosphate group, and that's the same for DNA and RNA. Now, I said we were going to look at the nitrogen bases because there are several different examples. So for nitrogen bases, they're divided up into their size. Pyrimidines are what we call the one-ringed nitrogen bases. And for one ring, those would be cytosine, which we abbreviate with a C, thiamine, and uracil. And you can see the abbreviations there. Now this picture will show you purines and pyrimidines. So with the pyrimidines, this is what we meant by one ring. Notice that they're smaller. The purines, which we're going to look at next, are 2 rings, and so they have one bonded to the other. Size-wise, they're about double the size. And there's only 2 examples of purines, which are the 2-ringed nitrogen bases. And we have adenine and guanine. As far as DNA, it uses thiamine, and it uses adenine, guanine, and cytosine. So DNA uses 2 of the pyrimidines, and it uses both of the purines. RNA, it uses adenine, it also uses uracil, it uses guanine, and cytosine. So you notice the difference here is between thiamine and uracil. DNA uses the thiamine, RNA uses the uracil, but all of the other nitrogen bases, both of them use. Now, we also said that one of those pieces to a nucleotide was a sugar, and so we need to know that for DNA, the sugar is called deoxyribose, and for RNA, the sugar is just a ribose sugar. And that's what the first letter of each of their abbreviated initials stands for, is the deoxyribo, and then Na for nucleic acid, and R stands for ribo, and then nucleic acid. And you can see a picture of them here. They are actually very similar. The spot in which they're different is here where it's highlighted in red. And you can see that the ribose sugar has an oxygen and then a hydrogen bonded to it, whereas in the deoxyribose it doesn't have that oxygen, and that's where the deoxy part comes in. It's a ribose sugar except for one oxygen. The shape and the structure of DNA, we hear about DNA more often, so many people know that it's called a double helix, or people may have heard of twisted ladder before. So the double helix shape is this one. So there are two strands, and helix meaning that they have this twisted, they're twisted around each other. The backbone of the DNA is very important. When we're building DNA, it takes that phosphate group and it takes a sugar. And you notice the sugars go in opposite directions. So here the sugars are kind of pointing down. Here they're pointing up. So they're parallel to each other, but they're going in reverse order. So we have the phosphate and the sugar, and then another phosphate and another sugar, and phosphate and sugar, and the backbone just repeats phosphate, sugar over and over again. And those are the two parts that make up the backbone. Now the inside of your ladder is a little bit more tricky. This is where your differences in your DNA are going to come from between you and the person sitting next to you. Your nitrogen bases, the adenine, thymine, cytosine, guanine, those are where you're going to find, you're going to find those on the inside of your ladder. So if you think of this is like the rungs that you're going to be stepping on if you were using this as a ladder. So guanine and cytosine bond to each other, but they do that on the inside, so they kind of point inward toward each other. We also know that the strands are complementary, so there's a rule that said A will only ever pair with T and G only ever pairs with C. And that's just the rule. It's the base pairing rule. So if we have the strand ATT, GCC, TAC, and we know that that's half of the DNA, keeping in mind that DNA is double stranded, so we know one half. We don't know what the other half is. We want to find the complement strand. Now, as an example, this is what we're looking at. So you only are given half of the problem. You know the certain strand of letters, so you can use the base pairing rule. Whenever you have a G, for instance, the other side is going to have a C. And whenever you have a C, the other strand is a G. A only pairs with T, and T only ever pairs with A. So that rule really helps us to decide what the other strand is. If you haven't already, I would like you to take a second, maybe pause the video, and see if you can come up with the opposite strand, and I'll put it up here in just a second. Now, whether you paused or not, I'm going to move on, so if you don't have this finished, do a quick pause. But the complement strand is this. A pairs with T, the T's here. We'll have A's on the opposite side. We have C, G, G, A, T, G. So that is what your opposite strand would be. Now if we look really quickly at the shape and structure of RNA, you're going to notice it does have some differences. One of the first things you might notice is that it's just a single strand. There is no double side there. So single-stranded for RNA. And the other thing that makes it unique is that uracil. And we talked about that on a previous slide, that RNA doesn't use thiamine. The ribose sugar is also something unique because DNA uses the deoxyribose sugar and RNA just uses the ribose sugar.